Okay, hello everyone. So we're gonna start now here with Guillermo Carrasco and Robin Andeer. They're gonna talk about how Python drives the analysis of billions of DNA sequences.
Okay. Well, hello, welcome everybody. Well, first, thank you for being here and not being on the other talk. I hope that you don't regret it. And yeah, thank you as well for letting us be here. So my name is Guillermo, and this is Robin. And we're going to talk about how Python drives the analysis of billions of DNA sequences. It's not going to be a very technical talk. It's going to be more of a use case of how Python is used in life sciences. So a little bit about us. We do work in Science for Life Laboratory, which is a research center focused on health and environmental research located in Stockholm with another node in Uppsala. And one thing that we want to mention about us is that we have very different backgrounds, even though we are working on the same team or field. I'm a purely computer science guy. I didn't take any course on biology. I took my engineering in Barcelona, and Robin is a biotechnologist. We think that this is important to mention because on the recent years, these two fields has kind of been merging into one field, common field, something sometimes called bioinformatics or computational biology. Actually, if you think a little bit about it kind of you can find some analogies between how biology works and how computer works. So on the computer world, with using programming languages, you write functions that develop a specific tasks. You group them into executable code or software that then you run on hardware or on machines. In the gene world, you have these A, C, Ts, and Gs that compose the DNA. Long strings of these A, C, Ts, and Gs form genes that have a specific function on the body. And if you put them together, you group them in executable chromosomes that then you can run on cells. So you can find this kind of analogy. So in our case, in SciLife Lab, which is an abbreviation for Science for Life Laboratory, we use Python. And we are going to talk about how and what for we use Python in research. So one of the biggest challenges that we face as bioinformaticians is the exponential growth of data that we have been experiencing in the last 3-4 years, let's say. If you have ever gone to any talk of bioinformatics or so, you will always see this chart. So the big problem is that the cost of sequences has dropped down a lot. So for the same amount of money, you can get a lot more data and a lot more information. So just to get an idea of what's the data flow that we use, everything starts with a sample that is sent to us either by researchers or doctors. Then we put this sample into the sequencer machine. That basically what it does is digitize this information into something understandable by computers. This information goes to our local servers where we do some processing of this digitized information. Basically, we convert this information into the strings of A, C, Ts, and Gs that then we run the analysis on. From the local servers, the data has to travel to 2 places. Basically, we need to send the data for a long-term storage facility because, well, we need to back up the data. And the process data has to go also to the computing cluster where we run the proper analysis. Very superficial level, the analysis consists on, well, first a quality control check of the data, and then we take these sequences that comes from the samples and we compare them with a standardized reference genome or reference human genome, looking for variations that can pinpoint the cause of a disease, for example. It's a very superficial level. So a couple of years ago, all these steps were possible to do them manually. You had a poor guy doing all these transfers and starting analysis manually. But with the amount of data that we have today, this is kind of impossible because of the amount of projects and samples that we work with. So basically how it is done today, we developed a tool called taka that stands for Tool for the Automated Cleanup and Analysis. it's also a short DNA sequence if you take care of it. So basically, Taka is running on the background, would say as several cron jobs in our servers, and it's performing basically all the steps that I showed on the flowchart before. Yeah. So even if it's running on the background, it has been designed to be user-friendly and nice command line interface, so that bioinformaticians or people that is not used to use this tool can easily understand it. So for doing so, we used a combination of standard Python entry points and a framework for developing command line interfaces called clique. So this is partly how your setup.py will look like. You're basically defining the entry points key and the console scripts that will point to a module in your, or a function in your module. This will create an executable for you that you will run on the command line. Then you also define your subcommands that will nest under the top command, which is the script that Python has generated for you. So in this case, you will have taka, and then taka storage or taka analysis. This is the piece of the small piece of code that you need for building a command line interface with clique. So basically, you create a group for the main command. Then the important thing to notice here is that we are loading the subcommands dynamically. So you can build your tool dynamically loading subcommands from either this package or maybe plugins as well. Clique doesn't also only help you with that, it also will parse all your doc strings and construct a very nice help interface for your command line tool. So it will look like this. So it's parsing the command line, as you can see, the doc string, sorry, and you have a specific help for the options of this command and a help for the following nested subcommands. If you keep adding the -- help to the subcommands, you will get help for the other subcommands as well. So this is very useful for people that is just learning how to use the tool. As I said, like 2 slides before, one of the things that Taka is doing is starting the analysis on the computer server, on the computing cluster. So the other piece of software that runs this analysis is called the NGI pipeline. That stands for National Genomics Infrastructure Pipeline. We are not very good with the names. This is a brief schema of the architecture of how is this working. So we have some helpers. In this case, one of them could be Taka, that is helping to start the analysis, for example, sends messages to a trigger, which in this case would be a very simple Tornado web server. Basically, it's just getting HTTP requests telling it to start analysis. We have a small database to keep track of running jobs and analysis. And then we have the engine, which is the part of the code that actually does the analysis. It is a very common problem in bioinformatics that everyone tends to end up developing their own thing, so their own engines. So we did the same, but at least we try to do it in such a way that we can reuse others' code. So basically, we focused on this part, on the engine, and we designed this in such a way that just specifying on the configuration file of the tool what engine do you want to run for what part of the analysis, you can load different engines or engines. Yep. So, well, you also have to build a small interface for the engine, of course. So this would be the piece of code that is used for launching the analysis. You basically iterate over your projects and samples, and here is the important line of code. Only specifying QC and the configuration file, I'm loading the QC or a specific QC engine that stands for quality control, is the part that just checks that the data has the quality that you expect. Only if you change the QC for BP, from best practices, you are loading another engine or whatever is defined in the configuration file. Maybe it's the same for running the best practice analysis, which is the one that compares our sequences with the reference genome. So these 2 tools have been really useful for us. They have helped a lot on improving our workflow, and we have now the capacity of working with a lot of samples on projects. The result of this analysis are a bunch of files, maybe some report, that are okay to give to researchers. They know how to work with them, they know how to interpret them, but it's not the same, or it's not okay to give that to, for example, a doctor that doesn't know how to interpret these results. For that, you need a further processing and visualization of these results, and this is what Robin is going to talk about now. So.
Yes, so I'm Robin. I work in the same SciLife lab, but in a different facility called Clinical Genomics. We're kind of a spin-off from National Genomics Infrastructure that Guillermo mentioned. And we focus on samples that get delivered from hospitals, so it's actually patient samples that need to be diagnosed with something. Right, and our end users are then doctors, so it's not researchers. And this is something that we have to think about when we deliver the data, or deliver the test results, basically. So one of the long-term goals has been that the doctor shouldn't spend more than 5 minutes dealing with the test results before they make their clinical decisions. So before I get back to the analysis part, I just wanted to mention what kind of diseases that we are talking about specifically. So this is then patients, often very young patients, because these disorders are disorders that you're born with. You inherit them from your parent or your parents. And they often develop very early in life and give you severe consequences. And they're also 100% genetic, which makes DNA sequencing a very attractive tool to use when you try to diagnose them. So the current situation is basically that these patients get into the hospital systems, they meet a doctor, they get passed around to do some tests, they get sent home, they come back for more tests, get sent home again, meet some new doctors, and this goes on and on and on for maybe 15 years. And you never get to really a conclusive decision. What is causing the disorder? How do we treat it, etc. So what makes people and doctors really positive and very optimistic about DNA sequencing is that within 2 weeks, you can actually get to the cause of the disorder. And when you have the cause, then you can start to think about how to treat these patients. You can start to give advice to the families about future pregnancies and risks included. And you can also alleviate the healthcare system from a lot of this accumulated cost over 15 years. So basically, we talked about this reference genome. So the analysis part that we do on our DNA sequences basically we end up with a list of variations between the reference genome. So if we get a perfect match between our patient sample and the reference genome, that would mean that we have sequenced a healthy white male guy from Canada. And any difference from that would then be represented with variation and differences in A's, T's, and G's. And C's, I guess, yes. So basically what we delivered to the doctors in the beginning was pretty much the raw output of this analysis. And we delivered it in an Excel sheet. It's got thousands of rows, basically filtered down from millions of variants to thousands, but still there's a lot of information here. And in the beginning, this was probably pretty intuitive. It's a familiar interface that the doctors all know how to use. We don't have to explain too much. But from someone that came into the team after this point with some limited web development experience, I could see that this was kind of deeply flawed. We could do so much better pretty easily. And I think this is also one of the benefits to bioinformatics. We come in with very different backgrounds, and when we collaborate, we can come up with nice things. So this developed then into a web interface that we call Scout. And it's now the main delivery mechanism that we deliver test results to the doctors. And I think that we can boil it down to two major benefits. One of them is that it has allowed doctors to interpret the results without the need for a bioinformatician backing them up. And also it's allowed them to share the results much more easily between the colleagues, rather than sending an Excel sheet, emailing to each other. So the way it's implemented is with a MongoDB database in the backend. This has worked really well for us, basically because we use this server as purely visualization. when we load the data from the analysis into the database, we basically don't touch it anymore. We don't try to edit it or anything, because if someone comes back to us in a year from now, we have to load the same data and show that the decision, or the basis of the decision, was this, that the clinical decision was made on, and we can't change that too much, or at all. Right, and so Mongo has been really nice, just dumping data in and then taking it out again. The server is implemented in Flask. I don't have any strong decision why we chose Flask. It was mainly that it was already what we had experienced with in the past. And we're also using other third-party libraries like Jinja 2 for the templating, and also MongoEngine to build up kind of an interface to the backend database. And I should also mention, because we're dealing with patient data, that's very sensitive, so we have to at least think about the security, even though we're not security experts. But we try to limit the access to the interface by restricting IP addresses that get access to it. We enforce two-step authentication across all the user accounts, and we also encrypt all the data using SSL. Or the traffic, I guess, yeah. So this is actually what the interface looks like. This is where your doctor would log in, and then they would see all the test results. So here is an overview of a patient case. We also give them this list again, slightly remodeled, with all the variants. But now they have proper tools for filtering it and kind of viewing only the most relevant information. And then they can, right, so I should explain here also that you can expect, or we expect then that we can find one of these variants to be the disease-causing variant. So then you can go in and look at each of these different variants. And here you can see all the annotations that we've collected. But we try to do it, visualize it in a way that at the top you get summarized information, and then as you scroll down, you get more and more into the details. Yes. So then on to some a little more technical aspects. We built this interface, a previous version of it, that was heavily built around a JavaScript framework called Ember.js. So it was interesting to listening to Emil's talk before this. But this is basically what the code structure looked like. And we did do, right, so I should mention maybe why did we choose Ember.js. Basically, I wanted to learn Ember.js, and I saw my chance to do it. And no one really told me that maybe it was not the best idea. But basically what we're doing is, in this interface, we rendered everything on the client side. And when you have these huge tables with lots of data, it makes for a really sluggish performance if you try to do data binding, etc. So we refactored it quite a bit, and we came up with a more sane structure that has made it a lot more easily testable. We can iterate on it faster. Because we try to do everything we can in Python, and only when we have to, we kind of write these JavaScript components. And this is also very important for us, because the developers that we work with, they all know Python, but they don't all know JavaScript. So it's no longer just me developing this, maintaining this. I can now bring in more people to collaborate on it. And we also realized that we're making a tool for professionals. And if it just gets their work done, they don't really care about the fancy JavaScript stuff, I guess. Right, so concluding this, I think that this has, of course, been a huge improvement from delivering Excel files, but it's also made us realize what else we can do to the to make the doctor's work more easy, and how far we can take this analysis in the future, which I think is going to be a very key piece of the puzzle in how to spread this across Sweden and more hospitals that are starting to use DNA sequencing for diagnosis of disorders. Right, so wrapping up, we wanted to point out a few maybe more general conclusions. And one of them is that specifically as bioinformaticians, we are kind of expected to do a lot of things, to have some kind of statistical knowledge, to handle data flow and automation, visualization, etc., etc. And having then a unifying language like Python that we can do pretty much everything in has really been very beneficial to us. And also, one thing that I picked up from yesterday is that maybe open source isn't always so like an obvious thing if you work in a commercial company as it is, like I feel like when you work in academia and science. I would say that most of the key pieces of software in bioinformatics are open source, and it's kind of an obvious way or obvious thing. And we want to contribute back to the community as well at SciLife Lab. So one of the efforts that we have is to highlight all the open source tools that we build and maintain in our organization. So we built this portal where we highlight different projects. And you can visit it at opensource.scilifelab.se if you'd like to know more. And of course, all the tools that we talked about today, they're also open source. You can find them on Github as well. And with that, I just give some credit to the Noun project if you want to find icons for a presentation. Yeah, the slides can be found at this URL. I want to thank you for listening. Yep.
Questions?
Yes.
Thank you for the interesting presentation. You have mentioned that you talked about you have built your own pipeline system. Haven't you tried to use something like Luigi from Spotify, for example?
Yeah, not personally me, but someone in the team tried to, and actually build something on the top of Luigi for doing some kind of analysis as well. Just that doesn't work very well on our computing cluster. We are very limited by it, by the things that we can do there, basically. So you have to think that, for example, we have our own scheduler there, which is called Slurm, or Slarm, that takes care of distributing jobs and all this stuff. So putting that, or Luigi, or another scheduler on the top of this is difficult. I think that he got to the point that the tool was working, but only locally, like in one computer, which is not very useful. But yeah, we have tried several options.
Right, thank you. That was a fantastic talk. My question is, are you guys storing the data, the DNA data, into Mongo just raw, or are you guys encrypting the data before you put it in the database? And if you are, how are you doing that?
No, we're not encrypting anything before we're storing it in the database. I guess it's one thing that always comes up when we discuss features that we can add, and we're kind of limited in some ways. But we are relying on more like downstream security if we encrypt the data and we trust that the clinicians aren't leaving their laptops unguarded and so on.
Also, it's not the raw data that we're archiving or putting on the database, but the result of the analysis. The raw data, we just compress it and send it to a long-term storage facility in case that we need to rerun some analysis or something.
Yeah. So, I mean, I could say that it is a very difficult problem because all of this data is personally identifiable data. Kind of the nature of it is that. So it's, yeah, makes a lot of things very difficult.
Okay, if there are no more questions, please give a big round of applause.